second wave attack and apprehending the third attack. But the ray of hope is the entire scientific community worldwide has been doing their best in their laboratories. When a result, we have already got some vaccines. And requested them to go ahead. So I hope this seminar, this web having the title Circadian Clock in Cyanobacteria will be very much
হ্যাঁ পেয়েছি, এক সেকেন্ড।
হ্যাঁ, ওই মেলে তুমি গ্রান্ট করে দাও, আমরা আমরা খুলে দিচ্ছি। তাহলে আমরা মেল গ্রান্ট অ্যাক্সেস করে দাও।
কি আমি শুধু দেখতে পাচ্ছি তিনটে অপশন দিচ্ছে, এডিট, শেয়ার তো।
হ্যাঁ,
হ্যাঁ ওকে,
আর কি কি অপশন আছে?
জাস্ট শেয়ারই আছে এখানে। একটা আছে কি এডিটর, কমেন্টার আর ভিউয়ার, আই থিঙ্ক
এডিটর এডিটর এডিটর
শেয়ার। আমি দেবজানিকে করে
এডিটর।
দিয়েছি, আই থিঙ্ক
হ্যাঁ হ্যাঁ।
দেবজানি।
দেবযানী হয়ে গেছে, ঠিক আছে দেখছি আমি,
হ্যাঁ
দাঁড়া। ওকে, এসে গেছে আমার।
এসে গেছে?
হ্যাঁ এখন।
ডাউনলোড হচ্ছে দেবজানি?
ডাউনলোডের অপশন
দেবজানি?
হ্যাঁ হ্যাঁ, এক মিনিট এক মিনিট।
So I request all the participants to be a bit patient. We are facing some technical problems, but soon we will be able to resolve it. And in a way, it's a boon, because some of our students, since today is a working day, some of our students are having their classes in the morning. So they requested me that they'll be joining at 10 a.m. So in a way, it's good for them that they'll be able to see from the start of the presentation. So it's okay. Just I request all the participants to be a little bit patient. That's all.
এখানে অনেক পিকচার্স আছে কিন্তু হিট করছে, লোড হতে সময় লাগবে।
ও ঠিক আছে, লোড হতে সময় লাগুক, কোনো ব্যাপার নেই। সুপ্রতিমদারই বাড়ি ফিরতে দেরি হয়ে যাবে অনেক। দেবজানি, তুমি একটা জিনিস দেখো যে
এক মিনিট, কারোর অডিও অন হয়ে আছে, একটু অফ করো না।
তুমি একটা জিনিস, হ্যাঁ বলো সুপ্রতিমদা।
আমি ভাবছি আমি একবার ক্রোম থেকে বেরিয়ে একবার সাফারি থেকে আমি এক্সিট করে একবার আমি সাফারি থেকে ট্রাই করছি ঢোকার, যদি ওখান থেকে, কারণ ওখানে দেখছিলাম আমি শেয়ার করতে পারছি, তো আমি একবার লাস্ট ট্রাই দিচ্ছি ওটা দিয়ে।
হ্যাঁ 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 হ্যাঁ, অবশ্যই অবশ্যই করো।
ওকে, ওকে।
হুম, হুম। দেবজানি, ইন দ্য মিন টাইম তুমি দেখো প্রেজেন্টেশনটা খুলছে কিনা, কারণ সুপ্রতিমদার কিন্তু ম্যাকের ল্যাপটপ, এইখানে বানানোর জিনিস তোমার ল্যাপটপে খুলবে তো? একটু শেয়ারিং ও দেবজানি
আমি
প্রেজেন্টিং শুরু।
হ্যাঁ, আমি
ওকে।
প্রেজেন্ট করছি। এক মিনিট দাঁড়ান, হ্যাঁ। আহ, সবাই দেখতে পাচ্ছো?
হ্যাঁ পরিষ্কার দেখতে পাচ্ছে, তুমি ফুল স্ক্রিন করে দাও খালি।
হ্যাঁ, এক মিনিট, ঠিক আছে।
তাও সুপ্রতিমদা যদি নিজে পারে সেটা সব থেকে ভালো, কারণ ও কন্ট্রোল করতে পারবে স্লাইডটা। ও ইয়ে থেকে ট্রাই করছে, তুমি একটু বন্ধ রাখো তোমার প্রেজেন্টেশনটা। সুপ্রতিমদা না পারলে এটা খোলো, কারণ এটা তো তোমার দেখা যাচ্ছে, বোঝাই যাচ্ছে,
হ্যাঁ
অসুবিধা
হ্যাঁ,
নেই। একটু তোমারটা বন্ধ রাখো।
ওকে।
এই তো সুপ্রতিমদারটাও কিন্তু দেখা যাচ্ছে।
হ্যাঁ, আমার মনে হচ্ছে আমি ইয়ে দিয়ে সাফারি দিয়ে খুললে হতে হবে।
হ্যাঁ তোমারটাও তো দেখা যাচ্ছে এখানে প্রেজেন্টেশন।
হুম হুম।
ইউ আর প্রেজেন্টিং, এখানে আমি এটা খুলবো কি করে, পাওয়ার পয়েন্টটা খুলবো কি করে?
পাওয়ার
পাওয়ার
পয়েন্টটা ওপেন
পয়েন্টে
আছে
যাও।
তো? দাঁড়াও দাঁড়াও, পাওয়ার পয়েন্টটা ওপেন আছে তো?
হ্যাঁ, পাওয়ার পয়েন্টটা তো ওপেন আছে, কিন্তু
এই
হ্যাঁ,
তো দেখা
এইখানটায়
যাচ্ছে
ক্লিক
তো।
করে রাখো।
হ্যাঁ
দেখা
তোমার
যাচ্ছে।
দেখা যাচ্ছে, এর আগে দেখা যাচ্ছিল, হ্যাঁ এবার দেখা যাচ্ছে।
হ্যাঁ, এবার দেখা
ওকে,
এটাকে
ওকে,
একটু,
যাচ্ছে।
গ্রেট, গ্রেট, গ্রেট,
এটাকে
থ্যাংক
একটু,
ইউ।
হ্যাঁ 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 ইয়ে করে দাও তাহলেই হবে।
ফুল স্ক্রিন দিলে।
Full screen।
Visible to
এটাও,
everyone?
ইয়েস 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 অ্যাবসলিউটলি।
Okay. Sorry for the technical problem to everyone. It's a great opportunity to present my work in front of everyone today. So today I will be speaking on circadian clock of cyanobacteria. So for last few years, I have been working on type 3 secretion system, as Abhishek told that, yeah, I have means done on a lot of research on type 3 secretion system, and then I got this opportunity to work on circadian clock, which is a totally a different field, and I got excited and I just accepted the offer at hello. Hello?
শোনা যাচ্ছে, শোনা
Oh,
যাচ্ছে।
okay, okay.
হ্যাঁ
Yeah.
হ্যাঁ
And I
অ্যাবসলিউটলি।
accepted the offer to work on cyanobacteria, circadian clock of cyanobacteria. So, yeah, circadian clock is present within the humans and in all animal and plant life. So, today, but the simplest organism to study circadian clock is cyanobacteria. So first, I will give brief introductions on circadian, the different clocks which are present, circadian rhythms which are present in humans, and then I will go to the circadian clock of cyanobacteria. I will just discuss upon the three proteins, how they maintain the circadian clock of cyanobacteria, and then I will conclude my talk on different researches which are being going on circadian bacteria, circadian clock of cyanobacteria. So a circadian rhythm, it has been defined as a 24-hour cycle that are part of humans, part of body's internal clock, running background to carry out essential functions. The most well-known rhythm is sleep and wake cycle. We know that we have a fixed hour of sleep and wake cycle, because if we sleep less than that time, or yeah, then our body is not fit to do all activities, because we need that time span of sleep. Means if we sleep, means if our rhythm is for 6 hours of sleep, we need that 6 hours. If we wake up earlier, then throughout the day we will feel sleepy. And all these, and there are not only sleep awake cycle, there are also different hormonal cycles are there, body temperature cycles are there within the human body. And these rhythms are endogenous, which are generated from the body, but they can be adjusted to local environment, which or external cues, which we call Zidjibbers, and which include light, temperature. And recently in 2017, these Jeffrey C. Hall, Michael Rauschbach, and even Michael Young won the prestigious Nobel Prize for their circadian rhythm research, where they found that a gene which produces protein that builds up in the cell overnight, whereas during the day it get breaks down. So yeah, different the circadian rhythm. We have different circadian rhythm within the body, and mostly what I have highlighted in this slide is the sleep-wake cycle. Because if we travel now, from India to a different place, like if you travel to US, you can see that there is a day-night cycle change, and going there you will have this jet lag. You cannot adjust to that because your sleep-wake cycle is again affected. Then if you're working late night, again your sleep-wake cycle will be affected. Uh, Here we have the daylight saving timing, because the timing changes, and again means a lot of things which affect our sleep wake cycle. And it's not the sleep wake cycle which is present in human body. And this circadian rhythm is not only present in human body, these are present in all different animal kingdom, like protista, eubacteria, archaebacteria, animal and plant. So coming to the circadian rhythms which are present within the human are, if this I've shown this slide just within a 24-hour circadian rhythm. So you can see that uh, from noon, as we go towards the evening, our blood pressure and body temperature increases. And as we go towards the night, we know that melatonin, which induces sleep, it starts secreting around 9. And then slowly towards as we go towards the midnight, the body temperature and everything drops. You can see at the early morning, the body temperature is low. And again, when we proceed towards the morning, the melatonin secretion stops. So this is a circadian rhythm of a 24-hour within the human body. And this is not only, you can see there is body temperature, this melatonin, and even there are hormonal secretions as well. So there is a different rhythms which goes within the human body. So next slide. Just stop me. Okay, yeah. If you have any questions, I can't see where you're writing, but yeah, if anybody has any question, they can stop me and ask me. It's okay. So here I've shown that there are different circadian rhythms, and these rhythms, because they have this is called a period, because they are getting repeated after a time frame. You can see the cortisol, which is a hormone, it goes up, it shoots up, its synthesis or its concentration is up at certain point of time, and then again goes down and again goes up. So this is getting repeated, and we call this a periodic this is a periodic repetition. And you can see that all these don't have their maximum amplitude doesn't match with each other, and it doesn't have to be, because each has a different period and a different time frame where this period is being repeated. So yeah, you can see that at the cortisol, which is a hormone, they have a different periodic repetitions, whereas melatonin, again, they have different periodic repetitions during day and night. So this is during day, and this is the dark color is night, and this is the time. So these circadian rhythms are mainly controlled within the human body by the suprachiasmatic nucleus, which is in the brain. And as you can see, this is a very much complex to study within humans, because there are a lot of proteins involved, and then again neuronal study as well. So the simplest organism to study circadian rhythm is cyanobacteria. And that's why our lab works on, not only our labs, there are different labs which work on to study the circadian rhythm of cyanobacteria, because there are less proteins involved, and it's easy to study how these proteins interact. So coming to cyanobacterial clock protein. So cyanobacteria is a gram-negative aquatic and photosynthetic bacteria. It's also called blue-green algae, because if you see somewhere in the water turns green, which is we call algal bloom. So that is because of cyanobacteria. And cyanobacteria has three proteins, which forms the clock proteins, the three core proteins, which is called chi-C, chi-A, and sorry, it should be chi-B. I have mislabeled here. So chi-C, chi-A, and chi-B. And these three proteins interact in a rhythmic manner and maintains the circadian rhythm of the cyanobacteria. So first, I will give brief details about these three proteins, chi-C, chi-A, and chi-B. So chi-C, it's a hexamer, It's a 519 amino acids. So if we look into a monomer, it's consists of two domain. One is the C1 domain, one is the C2 domain. So C1, both these domains have ATPase activity. They break down ATP. So, but here it has a kinase domain because from the ATP, it transfers the phosphate group to a serine and threonine, and an autophosphatase because it by itself it removes the phosphate groups. And there is also a loop. You can see this is called the A loop. So these monomers they all associate with each other to form a hexamer functional chi. So here are the phosphorylation site. As I said, that this C2 domain, they break down the ATP and phosphorylate this serine and threonine. These are the residue sites, S431 and T432. So T432 gets phosphorylated first, and then S431 gets phosphorylated during this phosphorylation phase. Another most important thing is that, as you can see, that these two domains are being attached to each other with a linker, and this linker is very much flexible. So at some part of the time or some part of the day, these two are not attached to each other and remain separated. We call it unstacked. And during other part of the day and time, they come close to each other, and we call it stacked. So this is highly breathing and dynamic. So they can separate out from each means uh, becomes independent. No, not independent means they just loosen up and then again becomes tight. There is the other protein which I said is Chi-A. So it's in a dimer form and it's 284 amino acids. So this Chi-A, where it binds, the Chi-A mainly binds to these A loops. And when does it binds? So these white, you can see I have labeled this as serine and threonine sites where it gets phosphorylated. This deep blue color is the C2 domain, and this light blue color is the C1 domain. So mainly this Chi-A binds to these A loops here. And when does it bind? When these are not phosphorylated. So when these are not phosphorylated, only the Chi-A binds, and these two domains during that time are separate from each other. When these two, okay, I will proceed. Now here is another protein which I said I mislabeled. There is Chi-B. So Chi-B in its original form is a tetramer. And when it binds, you can see it binds when it becomes phosphorylated. When these two regions become, these two residues of serine and threonine become phosphorylated, you can see these A loops, they get tucked inside. And these two domains come close to each other, we call it stacked. When these two domains get stacked and when these are phosphorylated, there is the Chi-B binding site here, which get exposed. And then Chi-B, from tetramer, it becomes monomer, and then it binds to these loops. Now, how does this rhythm takes place? So during the daytime, or we call it subjective 12 hours day or 12 hours night. So during the day or during certain, or if it's not, I would say, for now I'm seeing a daytime. So during the daytime, Chi-C remains unstacked because these are not phosphorylated and these are separate from each other. So it's unstacked, Chi-A is active, and it comes and binds to these A loops. During this time, both C1 and C2 domain, the ATPase activity is going on. As the day proceeds, you can see the A loops are exposed so that Chi-A can bind. As the day proceeds, and during the daytime, this Chi-B is also remains in a tetramer form. It is not in a monomer form. So when it's not binding to C1, it's always in a tetramer form. So as day proceeds, you can see this, since the ATPase activity is going on, first the threonine gets phosphorylated, and then during the midday or as it evening, and it proceeds towards the evening, both of these gets phosphorylated. As these both residues, serine and threonine, get phosphorylated, Chi-A falls off because these loops are tucked inside. Now what happens when these two gets phosphorylated, as I said earlier, these two domains come close to each other, as a result of which these binding loops get exposed. When this binding loops gets exposed, Chi-B now starts binding to this region. It becomes monomer and starts binding to this region. And as this is early evening and during late night, what happens? This six hexamer, they have six binding sites for Chi-B, and all six binding sites are means get the Chi-B binds to this all six binding sites. And what happens? This Chi-A now gets sequestered. Means Chi-B now is able to sequester this Chi-A, and now Chi-A binds to Chi-B here. But as the night proceeds, you can see it starts getting dephosphorylated, because you can see this red one has become white, which means it has lost the phosphate group. So as the night proceeds, it becomes dephosphorylated, and slowly what happens as the day proceeds, it's like a circle, it becomes like this. So during the daytime, again, what happens when these two gets dephosphorylated, it becomes white. Chi-B falls off. Chi-B falls off, as a result of which these two domains get separated and the A loops gets out, and again Chi-A binds. So it goes off like a cycle, which is totally a rhythm of 24-hour day and 24-hour night. So this is this main core clock proteins within the cyanobacteria, and you can see how this three proteins interact with each other uh, during the day and night, and maintains the rhythm within the cyanobacteria. Now, how did they study this rhythm? So, as I was saying, that this was an in vivo technique which is being studied. Now, one cool and very interesting thing, that when you mix these three proteins in a test tube, so it's so it's so cool, I would say, and interesting, that when you mix these three proteins in a test tube, like by adding ATP and magnesium chloride at any time of the day or any time of the night, that's why we're saying subjective 12 hours. And what happens, the ratio should be like this, because it has been experimentally verified, that chi A should be around 1.2 micromolar, chi B 3.5 micromolar, chi C 3.5 micromolar. And if you leave it in a test tube, what happens? You take time points at different time points. Now here I'm showing a gel of only chi C. When you add these three proteins, chi C is, as I said, 519 amino acids. So it remains at the top, whereas chi B, because since it's a higher molecular weight, so we run a 7.5% gel where both chi A and chi B is not visible, only chi C is visible. So at different time points, if you add in a test tube and take out aliquots and run a gel, you can see two forms of chi C. One is a phosphorylated form and one is the non-phosphorylated form, where I have shown here. So as the time proceeds and when you add these three proteins, you can see the band of phosphorylated getting darker, and slowly after some time, it's again the non-phosphorylated chi C becomes darker. Again, at some point, the phosphorylated gets darker, and again at the other point, the dephosphorylated get darker. So when you analyze these gels, the intensity, the band intensity, you could see the like this type. So if you add the ratio of phosphorylated chi C with ours, you can see the ratio of phosphorylated chi C slowly getting increased and then falling off, slowly getting increased and then falling off. So this is, you can see a period of chi C phosphorylation. So this is the circadian, this is a rhythm which even if you add these three protein outside cyanobacteria, so you can see how interesting these three proteins are, that they can maintain even a rhythm of their own binding, association, and dissociation, which is really interesting and fascinating to study this three clock proteins outside cyanobacteria. Because if you means it has been observed that if you change these concentrations, you can change this rhythmic pattern. This period become faster, because you can see it's close to 24 hours, it will shorten, or it will get delayed, and it will take more time. So that's why people are mostly studying this circadian rhythm within the cyanobacteria, because you can just generate the rhythm just by adding these three proteins in a test tube. However, you can see that
Supratinda,
yes,
1 minute. Is it a gel or is it a Western blot?
it's a gel. You
It's
don't
a
have to
gel.
do Western blot. Yeah,
Okay, okay. Thank
sorry. So now our lab designed a new technique, because you can see sometimes this gel might not run well if you are not lucky, and it becomes messy. And again, you have to manually take time points and run on the gel. So our lab designed a technique by through fluorescence anisotropy. So fluorescence anisotropy, what they did, that there is a light source and a polarizer, which makes unidirectional. And what happens, as you have seen, Chi-B is a small molecule. So this is Joel in our lab. So he developed this technique. So what he did, he added a fluorophore to Chi-B, which is Chi-B, is very small. So he added a fluorophore to Chi-B. So when Chi-B is free, it's not binding to Chi-C. It's rotating very fast. But when it's binding to you can see the molecular weight of chi c and then chi a, it becomes so big, so it cannot tumble so fast. So its rotation is slow. So when you're studying this fluorescence anisotropy in a plate in a fluorimeter, so what you will see, oh, I'm sorry. So when you're studying this, only when this chi b is present, it's not binding to chi c, you will get a depolarized light and with low anisotropy. So the anisotropy will be value will be very low. But when it's binding to this complex, chi b is binding to chi c and then chi a, the polarization will be it will be a polarized light and it will be of higher anisotropy. So what we studied that during in a fluorometer, when you study the anisotropy versus time, if you add these three proteins, like what I said, so I will show here, again, you're adding these three proteins in a plate, in a simple plate, you're putting this plate in a fluorometer and just leaving it. And with time, you're studying the fluorescence. So you can see this cool thing, the fluorescence anisotropy going up, down, without taking any time points or anything. So this was a new technique, and now being means most of the labs are now following this technique, because there is no manual labor or gel running. You just add these three proteins, leave in the fluorometer for 5 days, 6 days, and you will see slowly this rhythm of the fluorescence anisotropy. So what does this peak and this down means? So during this peak, you know, since the chi b is binding to a chi c, which is phosphorylated, and these two are stacked. So this peak means that the chi b is binding to chi c. Either it's by its own or it has sequestered chi a. And during this trough, you know that it's only the chi b is not binding to chi c, so it's just alone by itself, and this part mimics this form of chi c, where chi c is dephosphorylated and chi a is binding. So you can see by just using fluorescence anisotropy, you can study the rhythm of cyanobacteria of these three proteins in vitro.
স্যার একটা কোয়েশ্চেন করব।
Yes.
স্যার বলছি যে যখন কোনো প্যাথোজেন হিউম্যান বডিতে ঢুকলো, তো স্যার এবার সার্কেডিয়ান রিদমটা হিউম্যান বডির কি চেঞ্জ হবে না সেম থাকবে, বা সাইনোব্যাকটেরিয়া তো নিজের প্রোটিনগুলোই তৈরি করবে হিউম্যান বডিতে, তাহলে কি হবে এই ক্ষেত্রে?
সায়ানোব্যাকটেরিয়া হিউম্যান বডিকে ইনফেক্ট করে না, ফার্স্ট অফ অল
মানে কোনো প্যাথোজেন
নর্মালি শিওর আমি বলতে পারবো না, বিকজ উই মানে আমরা এই জিনিসটাকে প্যাথোজেন হিসেবে স্টাডি করি না। বাট হ্যাঁ, রিসার্চ পেপার্স নাও, লাস্ট দিনকে একটা রিসার্চ পেপার দেখলাম, ব্যাসিলাস সাবটিলাস অর সামথিং, মানে ইট সামটাইমস ইট মডিফাইজ হিউম্যান রিদম অর ইট কোপস আপ উইথ হিউম্যান রিদম, বিকজ এটা আমি দেখছিলাম যে ইভেন কি malarial parasites, they are also affecting human rhythm or getting adapted to a human rhythm, something like that. Because researchers are there, means if you search, means bacillus or even parasite, malarial parasite, the human proteins যেগুলো circadian proteins সাথে their কিছুটা ভাবে their rhythm কে affect করছে, means what I know, but yeah, surely আমি বলতে পারবো না।
থ্যাংক ইউ স্যার, থ্যাংক ইউ।
Yeah, if you see that the same both the experiments were run simultaneously. If you see that this is fluorescence anisotropy and this is running gel, and you can see, if you're not lucky, your gel may look like this, where you cannot differentiate between phosphorylated and dephosphorylated chi c, because maybe you didn't make the gel well. But here you can see that just adding into a plate, it does give you a good rhythm and a good output of your results. So yeah, again, I'm plotting means this ratio of phosphorylated chi c from this gel, and you can see definitely it matches with this rhythm what you see in the plate. So yeah, definitely people are now opting for this technique more than this running phosphorylated dephosphorylated chi c on gels. Now, I have shown you that these proteins interact in vitro, but the main question is, how do they regulate the circadian rhythm, or how do they regulate the gene expression? So there are again two proteins which play a role, and these are called output proteins. So there are two output proteins. One is called SAS-A and one is called KIK-A. So what does these two proteins do? So there is a transcription regulator called RPAA. So when SASE gets phosphorylated, it activates RPA by transferring its phosphate group to RPAA. Now RPAA binds and there is transcription of DNA. KK, when it binds to RPAA, it removes the phosphate group and it inhibits RPAA. Now, how these three proteins fit into the Chi-A, Chi-B, and Chi-C. So you can see that RPA can regulate so many gene activity of these genes and functioning of different activities of cyanobacteria. So how does that these three protein fit into this model? So what happens during as it goes from this single phosphorylated to double phosphorylated, the SASA binds to Chi-C and gets phosphorylated. But when Chi-B starts binding, it knocks off SASA. So as Chi-B starts binding, SASA gets phosphorylated. It transfers phosphates from Chi-C to on itself. And when Chi-B binds, it knocks off SASA. Now SASA gets phosphorylated and it transfers the phosphate group to RPAA.
সুপ্রতিম দা একটু একটু ওয়েট করুন, কেউ একজন অডিওটা অন হয়ে আছে। কন্টিনিউ কন্টিনিউ।
Okay, so here you can see that SASA gets phosphorylated and now it interacts with RPAA and it phosphorylates RPAA and starts gene expression. During the daytime, what happens? KIK A is present here within the, I would say, cyanobacteria free, and it always keeps RPAA in an what to say, not in a dominant form, because RPA cannot bind because it always dephosphorylates RPA, and RPA cannot bind to the gene and cannot transcribe. So, but as you say, during the night time, what happens? KK starts binding to Chi-B, so it cannot anymore inhibit RPA phosphorylation. Whereas SASE during the night time, it's phosphorylating RPA, as a result of which RPA is now can bind to gene and can start transcription of various genes. So you can see how this, from this, the entire model, how this circadian rhythm of this Chi ABC and along with SASE KK, they maintains the gene expression of various genes within the cyanobacteria. So,
Excuse me.
yeah.
Can you please repeat the Sase and Kike, the thing about the timing during the day and night? I just missed that point.
So during this part, when it starts getting phosphorylated, the SASE gets bound to this, before Chi-B binds, the SASE gets bound to Chi-C and starts phosphorylating itself. When Chi-B starts binding, it knocks off SASE. So it is seen that only one SASE can bind to this Chi-C. It's not like that Chi-B can bind to 6 Chi-B can bind to 6 Chi-C. It is still now observed, though research is going on, but still now observed that only one SASE can bind to Chi-C. And this SASE gets phosphorylated when it binds to Chi-C. Now, when Chi-B starts binding to this complex, it knocks off SASE. Now this phosphorylated SASE is free, and now it binds to RPAA and transfers its phosphate group. During the daytime, KIK A is free, and it keeps RPA in an inhibited form because it removes the phosphate group, as I said. But during nighttime, you can see that KIK A is being sequestered with Chi-B. Chi-B can sequester both KIK A and Chi-A. As KIK A is getting sequestered, now SASE is free, means now RPA becomes free and it can bind to SASE. And now SASE, what does it? Transfers phosphate group to RPAA. Now RPA is active and can start gene expression of different genes. So it's mainly through this circadian, means this phosphorylation, dephosphorylation of Chi-C, and then with Chi-A, Chi-B, along with SASE KK, it can regulate the phosphorylation, dephosphorylation of RPAA, which is responsible of transcription of various genes.
Okay, sir. Thank you.
Okay, now before going to this slide, I would like to stop here, and I would say that, yeah, in vitro, as I said, that you can see rhythms of Chi-C phosphorylation, dephosphorylation. This lab has moved to such an extent. Now, recently, from our lab, paper has been communicated, and it's in a communication step that since RPA, you know that RPA binds to a DNA. So now it has been seen that if you put that DNA within the tube or within the plate, you can see, and put a fluorophore on the DNA, you can see even the DNA binding to the RPAA and falling off from RPA. So you can see even the binding of DNA to the RPA, there is also rhythm. So yeah, research means we have means our lab has successfully moved to that level of research where you can see binding of the binding of DNA to RPA in a rhythmic fashion. And now we are being discussing, or it means through collaboration, how we can study the in vitro, because we know that we can do protein synthesis. Means there are translational systems which we can external translation systems, so how we can add that and see how we can regulate this how we can study this translation of proteins, or proteins produce, means the rhythmic activity of the proteins within cell. So yeah, we have through this fluorescence technique and everything, we have moved forward much forward in studying this system. And there are also many things which are not answered. So I will just, in a concluding slide, I will briefly highlight those research areas which are still means which are questions which we are still trying to address. So this is the part which I discussed about the circadian rhythm of cyanobacteria. Now, one more interesting thing, which is which again falls here. So I don't know how much you are more acquainted with the term of metamorphic protein. So you can see here Chi-B. So I've shown with the different, the question arises why it's circle here and why it's rectangle here. If a protein means it should have the same conformation. So one cool interesting thing is that Chi-B, when in a tetramer form, it has a different conformation or a different fold. Whereas when it becomes monomer, it adopts a different fold, which is against what I would say, I'm forgetting the rule, because you know that one sequence, one fold. You cannot have a amino acid sequence, a secondary structure, but having two different folds. So the
Yes, sir.
protein,
It's Unfinson's dogma, is it, sir?
yeah. So that's why this Chi-B is now interesting, because it's showing two different folds, but the same protein sequence. So yeah, the prevailing dogma is that in a structural biology is one sequence, one fold, whereas in a metamorphic proteins, you can have different means the same amino acid sequence, but it's giving a different secondary structure at different chemical environment. And yes, there are, we till now it's 20 metamorphic proteins which we know, but definitely we are means research is being going on to study means methods to study or predict more metamorphic proteins, because secondary structural prediction servers will just give you one form, but when you do experiments like NMR and different techniques, you will come to even, excuse me, even CT. There it shows up that the protein has two different conformations, whereas when you're doing a secondary structure prediction and everything, it only gives you one prediction structure. So that's why research is being going on to develop this prediction server, so that they can also even predict that this protein is metamorphic or not. So there are more metamorphic proteins like this C terminal of RFAH. You can see the same sequence, it's having two different fold. Then it's lymphotactin. Again, it's the beta sheets turns into an helical, and then it's a Chi-B. So I'm getting what transition takes place within the Chi-B. So the Chi-B, because we know it's a ground state, we call it a ground state fold. And then when it binds to Chi-C, we call it a fold switch fold. So in the ground state fold, you can see that the protein has something called beta, alpha, beta, and then beta, 2 alpha, and 1 beta. But when it switches its fold, because this is forms in a tetrameric, but when it becomes monomer, you can see the fold switch which is taking place. This part of the protein remains same, beta, alpha, beta. But here you can see this has switched. From beta, alpha, alpha, beta, it became alpha, beta, beta, alpha. And here I've shown that the same secondary structure, one, which is the ground state fold, you can see the change. Here is beta, 2 alpha, and then beta. And now through NMR, it has been seen alpha, 2 beta, and then alpha. So this is a cool thing which has been seen for Chi-B. And it's the only metamorphic protein till now known for circadian clocks. But the big question is, how does this metamorphosis taking place? Means how Chi-B is from a ground state fold becoming a monomer, and then monomer switching its fold. So it's means we are now studying on that, because if we can break that mechanism, that will be a really an important thing to study. So yeah, how does this fold switching occur? So Kaibi has many prolines, and it is being means it is being hypothesized that maybe these prolines through cis trans, because you know the prolines has a trans state and a cis state, maybe through cis trans isomerization, this fold switching of Kaibi is taking place. But however, it's still not established, and this is the hypothesis which we are working on. And yeah, this is the main another aspect our lab is trying to study, that how this fold switching is taking place, because till now there is no certain pathways or anything known for metamorphic protein fold switching, because we know that okay, this part is fold switching, but yeah, a definite path, at least for Kaibi, it's not known, and we are trying to uh, see that through our experimental as well as through what to say computational techniques. So yes, I just covered most of my points, and now I'm just coming to the conclusion slide. So what is the ongoing research on circadian rhythm going on means on cyanobacteria? So what is that? What makes the clock so slow? Because you can see that it takes half of the 12 hours day to phosphorylate just 6 Chi molecules, and then dephosphorylate. Means why it's not faster means you can see other ATPase activities, they were so faster, but for Chi its ATPase activity is slow that it produces only 12 ATP molecules throughout the day. So yeah, research has been going on why this clock is so slow, because there is an intricate network between the C1 and C2 domains that is getting regulated, which we still don't know. Then how does stacking between these two domains of Chi-C occur? The mechanism, the intricate network, we still don't know, because how these two domains stack, what signals it's one domain is sending to the other domain, it's still a mystery. So now the research has been going on that people you can mimic these phosphorylation state by doing mutations, and people are trying to do what we have come to know, cryo-electron microscopy, to see these different phosphorylation states, to see how these amino acids, or how if there is any network or a pattern taking place between these two domains. Again, binding of 6 Chi B molecules to Chi-C. Again, you can see this. There are different models, because first of all, Chi-B is changing its structure, and again, this binding of Chi-B to Chi-C, is it an allosteric model or a different kind of model? Because all 6 Chi B molecules cannot bind together to Chi-C. So again, if binding of one is facilitating the other, and if so, how this model is taking place. Then is the temperature compensation. Temperature compensation, even if you increase or decrease the temperature, the activity remains, it's tried to maintain its Q10 value, which I haven't gone into details. So yeah, it tried to maintain in Q10 value 1. So yeah, temperature compensation is also an aspect of means a research which is being going on this circadian rhythm of cyanobacteria. And lastly, which I am dealing with, is now doing NMR and everything on fold switching mechanism of Chi-B. Means I'm trying to do different HD exchanges, HD exchange study of Chi-B, to know which domains are flexible, which domain is rigid, and using different denaturations like urea and increasing concentration of urea to know which domain is dynamic, which can loosen up and help Chi-B to fold switch. So currently my research area is to study the fold switching mechanism of metamorphic protein of Chi-B. So with this, I would like to conclude my talk. I just gave an overview of the circadian clock of cyanobacteria, as well as what research has been going on. And yeah, I like to thank my professor Andy Levang, and here are other colleagues of mine who are working on this cyanobacterial clock. So yeah, I like to thank you all for listening my talk, and I'm open to questions.
On behalf of all the participants and the organizing committee, I once again thank Dr. Shubhrotin Dey for this wonderful lecture. Now we have some questions. Let me read it out for Dr. Shubhrotin Dey. Dr. Raja Ghosh asked, what was the fluorophore used in the anisotropy experiments?
6IF.
Can you please repeat, Shubhrotin Dey?
6 IAF. I do acetamide chloride. I don't know, but yeah, I will.
Yeah, that's okay. That's not a problem. Okay. So, Shayan Vishya, she is a student of ours. He asked how the Chi-C protein sense the day and night time.
Yeah, that's a good question. That's why I'm saying that I said that it's day and night, but I'm saying it's subjective day and subjective night, because I told you in a test tube, if you mix it at any time of the day and any time of the night, it just maintains that 12-hour rhythm, that after 12 hours it will dephosphorylate, and after 12 hours. So yeah, comparing day and night to means comparing day and night and Kai-C phosphorylation dephosphorylation, that link I don't know, but yeah, subjective 12 hours day means during this 12 hours it will dephosphorylate, and during this 12 hours it will phosphorylate by its own, and it's, yeah, means I cannot correlate that with day and night, but yeah, subjective 12 hours day and 12 hours night, we say like that.
Supratim, I have a question on this. Uh,
Yeah.
Other factors like temperature, pH, etc. Does these factors also affect this phosphorylation and dephosphorylation process?
Um, that's what I said. That temperature compensation is a term which is being used, which is called the Q10 value, and I haven't discussed much in details. Chi-C is temperature compensated because even if you change the temperature, it maintains its ATPase activity in a similar rate. So by that way, yes, this Chi-C is temperature compensated. The whole interaction of Chi-A, Chi-B, Chi-C is also temperature compensated. But now we are trying to study how this interaction of Chi-A, Chi-B, and Chi-C is temperature compensated. We know because studies have shown that the ATPase activity of Chi-C is temperature compensated, because if you increase or decrease 5 degrees, its ATPase activity remains the same. pH, I think pH is a factor, and there is a definite pH. Like we study two systems, one is Sinococcus and one is Thermophilus, and they need a different pH. Like for Sinococcus species, we use pH 8. For Thermophilus species, we use pH 7. So definitely pH and temperature does affect, but yeah, temperature it's compensated.
Okay, so we have another question from Shayan Bishash. He is asking that is that protein, I think he is referring to Chi-C only, phosphorylated by itself or by other proteins?
By itself, definitely by itself. It has autophosphorylation and auto dephosphorylation.
Autophosphorylation domain, right?
Yeah.
So we have many other questions as well. Shayon Dashi is also a student of ours. He is asking, is there any role of serotonin in circadian cycle?
I have no clue on that. People doesn't have serotonin, but yeah, quinone is there, and people are there, and quinone does have an effect. But yeah, I still don't know the total pathway of quinone. But yeah, quinone does play a role in circadian clock of cyanobacteria.
Okay. Rudrajit Mondal is asking, what causes potential shifts in the day and night times in the cyanobacteria?
Potential shifts. Sorry, I didn't get the question.
Rudrajit, can you please elaborate a little bit? Rudrajit,
মানে
are you there? হ্যাঁ বলো।
হ্যাঁ স্যার মানে যে টাইমটা ফিক্স হচ্ছে একটা 12 আওয়ার পিরিয়ড যেটা ডে টাইম, তো সেটা কি শিফট হয়ে যেতে পারে কোনো কিছু,
Huh,
মানে
that,
ডিসরাপ্ট হতে পারে
definitely
সাইকেলটা?
means. That's what I said. That means if you change the concentration, there is a fixed concentration. I said that 1.2 micromolar, 3.5 micromolar of 1.2 micromolar of Chi A, 3.5 micromolar of Chi B, and 3.5 micromolar of Chi C. If you tune those concentrations, you can see clocking moving faster or slower. So definitely you can tune that. And even recent papers have come that even a single amino acid mutation in Chi C can make the clock 6.6 days. Means after 6.6 days you're going to see a rhythm. So yeah, definitely you can tune that clock in a way of changing protein concentrations, mutating certain residues. By that way you can definitely change the clock.
Okay. Shomodip Roy, he is
ওকে
asking,
স্যার।
what is the significance of sequestration of Chi-A at late night? The significance of
It's
sequestration of Chi-A.
Yeah, because it's sequesters Chi-A, so that Chi-A is not, because the pool of Chi-A is less, and because it has been seen that if Chi-A is more, then the clocks gets faster, because there is free Chi-A, and as soon as the loop starts, it will start binding. So yeah, if you increase the concentration of Chi-A, there will be more free Chi-A available, and it will start means there is a certain lag phase, means slowness of the clock. But if there is more concentration of Chi-A is always available in active form, it will just bind to A molecules as soon as it get dephosphorylated. And it is being seen that increasing the concentration of Chi-A makes the clocks faster. So definitely sequestration of Chi-A concentration is required to maintain that 24-hour rhythm.
Shomodip De is again asking, what significance does cyanobacterial clock poses on its overall metabolic activity?
Yeah, definitely there are because recent papers are being shown that it affects glucose metabolism is there. And yeah, if a recent paper came out in Cell Reports where they have seen that different metabolites in lipid molecules or in a certain droplet molecules changes with this circadian phase of cyanobacteria. Yeah, definitely it has impact on metabolism. But yeah, we don't go to that metabolism because we are still focusing on chi A, chi B, and chi C. So there are labs which are definitely working on the metabolism pathway of cyanobacteria and trying to correlate with this chi A, chi B, and chi C.
Okay. Tamanna, she is asking, are the human homologs of this chi protein discovered? Human homologs of
I
chi
would say
protein.
Uh, human proteins are totally different. Means I would say there is no homolog, but there are proteins like per proteins. I'm not very much acquainted with the proteins in human. I always forget them because there are collaborators which work on human proteins, but I totally can't recall now. But there are PER, I think per proteins they call, or perk proteins. I'm not sure, but I won't say homologs. But yeah, there are proteins which behave similarly like that of what you see in cyanobacteria. That's why people try to study cyanobacterial clock as a simple mechanism.
Okay. Shauradip Shil, he is asking, as the cycle involves ATP, so it must be dependent upon the concentration of ATP. So if the ATP concentration is low, how does it affect the cycle?
Yeah, if the ATP concentration is low, the cycle will be definitely slowed down because you can change the I think cycle by using different concentrations of ATP ADP ratio. So definitely there will be slowing of clock. Yeah, still I think this is a good avenue we are trying to study the different changing the ratio of ATP and ADP and to study how the clock how chi means how chi C behaves. But yeah, to my understanding, it might slow down the clock.
Dr. Raja Ghosh is asking, can cyanobacteria occur naturally in groundwater aquifers? Groundwater
Pardon?
aquifers. Can cyanobacteria occur naturally in groundwater aquifers?
I am not sure about this, but I know it's always in fresh water because we call this blue-green algae. Means we have seen
Uh,
everywhere that whenever. So yeah, definitely it's fresh water, but I'm not sure about. Means I'm not sure about this. How to answer this question because yeah, I don't know.
it occurs in soil, in depth of soil. We could see that, but I don't know about groundwater aquifers. Okay, so that's all we have. I have a last question for you, Shubhatimda. Do you presume that there are many more proteins which might not be discovered till that working in this pathway of circadian rhythm of cyanobacteria?
Yeah, definitely because one of our collaborators is coming up with a new protein they have observed. However, they don't know where it will fit. But yeah, they have through pull down assay they have seen that protein. They are trying to characterize that protein, but yeah, they were presenting to us, and it's difficult to fit that protein in current model. But maybe, but still, because this model is always changing, as you know, if different proteins come. Because as I said, these studying this period and phase when this protein is coming and binding and when it's getting off, when chi b means studying that particular phase or particular that period, it's still under means the research is going on that. So yeah, maybe there are proteins which binding or affecting this clock is still undiscovered, but the core proteins like chi a chi b chi c will remain the same. Maybe it will be like something like sasa or kk, which are output proteins, but I think the input protein, the main core proteins will remain the same.
Okay. So Tamanna, she is giving a suggestion. She is saying that if chi-like proteins are present in human, then could those proteins be targeted for people suffering from sleeping disorders?
I would say that human system is very much complex. I shouldn't comment on that, but yeah, definitely sleep disorder studies been going on, and as I said that if you go to humans, definitely it gets complex because there, as I said, there is suprachiasmatic nucleus, and it's controlled by supra, and there are again neuronal studies which been going on, the protein studies. So it's hard to say like that way, but yeah, means means the clock proteins which are present in humans, they definitely regulate the sleep cycle of humans.
হ্যাঁ, কারণ ম্যালেরিয়ার মশা এক সময় মধ্যরাতে কামড়াতো, এখন কিন্তু সকালবেলার দিকে কামড়াচ্ছে, সব ডাক্তাররা সেটাই বলছে। So Tamanna, yeah, you could think about this. So that's all. We don't have any more questions. Yeah, Shopnil Roy again asked one question. Can circadian rhythm be altered in cyanobacteria in any way? I
Yeah,
mean, a
that's what.
bacteria as simple as cyanobacteria, can that be affected by heavy alterations? I mean,
What
just he is asking whether it can be altered or not in
Yeah, as I said
cyanobacteria.
earlier, that even if you change the concentration of these three proteins, Chi A, Chi B, Chi C, you can definitely change the circadian pattern or the how fast or how slow the clock goes, because that's the big tool in our hand to study this this system, because we can modulate the system, and even there are papers where you can just, it is seen that just changing one amino acid of Chi C, you can tune this clock. The period becomes from 24 hours to 6.6 days, means which is means that's totally means a different thing, means. So yeah, there are studies you can definitely modulate. That's why people try to study cyanobacterial clock, because easily you can modulate this or tune this clock by changing different things.
Okay. Supratinda, is there any other system where only three core proteins is controlling the entire circadian clock? Is there any other system like that?
As far as I know, still now is circadian. I mean cyanobacteria. There are even homologs where only two protein now. There are bacteria where only Chi A and Chi B, means they are seeing that homologs of Chi A and Chi B are responsible, and even not, sorry, Chi B and Chi C, even Chi A is not required. So till now I know of cyanobacteria. For others, because recently for Bacillus subtilis they have discovered a similar clock, but other than cyanobacteria, it's not coming to my mind now.
Okay, there's a last question from Joy Ghosh. I couldn't understand the question. RPA
Yeah.
depends on what without transcription process? Probably is asking that other than transcription, what regulates the expression of RPA or something?
Uh, that I would say I don't know, because that's again a different aspect of studying RPA, because that's again down regulation pathway. So yeah, I don't have a answer, means confirm means answer to that.
Okay, so with this, we again thank you, Dr. Shupratim De, for his wonderful lecture.
This was my
And
privilege. Thank you so
yeah,
much for
and
being.
yeah, and we will meet soon, not virtually, but in real time. And now I would request the convener of this webinar, Ms. Debjani Mondol, to deliver his valedictory speech and conclude this webinar.
Okay. Oh, again there is a question. Madhushri Chakraborty. She asked that, Sir, melatonin is said to be our biological clock. Has it any connection with this protein? ওটা তো আগেই
Uh,
লেখা।
Melatonin, no, I that's what I said. Melatonin, it cannot be correlated to cyanobacteria, but yeah, there is a small molecule which I called said that it's quinone, and it's being studied that yeah, quinone can affect a cyanobacterial clock protein. So similar to melatonin, I would say here it's quinone.
Yeah, so I just lost my connection a little bit. Again, I see one more question. Sir, melatonin is said to be our biological clock. Has
I
it
just
ওইটাই
any?
আমি গেট করেছি।
Okay, oh, you just read that. Okay,
হ্যাঁ
I
হ্যাঁ
missed
হ্যাঁ
it. Okay, so Devjani, you can conclude
হ্যাঁ
now
হ্যাঁ হ্যাঁ
with your
হ্যাঁ হ্যাঁ।
validity. Okay.
Okay, good morning everyone. Over the last 1 hour, we have learned a lot in this academic session. Our esteemed speaker, Dr. Shupratim De, has delivered his lecture on circadian rhythm of cyanobacteria. He has given a very brief yet informative talk, and that too in a very lucid manner. I'm thankful to you, sir, for enlightening us with your beautiful presentation. And you have given enough exposure to our students uh, about the current research scenario. You have enlightened them with the recent trends in biotechnology research. I'm pretty sure that all the students have received huge benefits from this academic session. This will no doubt be valuable as they go forward in life. Thank you once again, sir.
Thank you so
Thank
much.
you. Thank you. I also express my gratitude to our principal, sir, Dr. Shamsuzaman Ahmed, for his encouragement, advice, guidance, and support. He has always pushed us to excel. He motivates us to continue working as hard as we could. Thank you, sir. My sincere thanks to all the faculty members, research scholars, and students from such diverse fields. Without you, this event wouldn't have achieved success. Lastly, I would like to thank our organizing team, our joint convener, Dr. Abhishek Boshu, organizing secretary, Madam Shantani Boshu, and our technical expert, Sir Biplab Bishwas, for all their efforts. It's a true team success. Thank you, everyone. Thank you.
Okay, so thanks a lot. We can quit now. Okay.
Yes.
Thank you.
Thank you.
Thank you.
Supratinda, we'll meet in real time, not virtually.
Okay.
Yeah.
Bye, everybody.
Bye.
সুপ্রতিম দা ইন্ডিয়াতে এলে আমাদের কলেজে আসবেন চলে গেছেন
হ্যাঁ হ্যাঁ হ্যাঁ। সুপ্রতিমদার শ্বশুর বাড়ি কিন্তু এখানেই, মুর্শিদাবাদ।
ও ও আচ্ছা আচ্ছা তাহলে ডেফিনেটলি দেখা হবে আরে এখন সবাই জয়েন করছে ঠিক আছে তাহলে মিটিং এন্ড করি সবাই লিভ করতে
হ্যাঁ হ্যাঁ, এন্ড করে দাও, এন্ড করে দাও।
ওকে
বলে দি থ্যাঙ্কস আ লট সুপ্রতিমদাকে। প্রিন্সিপাল স্যারকে জানিয়ে দিও কিন্তু।
হ্যাঁ হ্যাঁ স্যার তো বেরিয়ে গেলেন তারপরে আর
স্যার মানে এটাও হতে পারে যে এত কিন্তু পুরো হয়ে গিয়েছিল, 100 ক্রস করে গিয়েছিল, তো স্যার
না না
হয়তো
ওই
ট্রাই
জন্যই
করছিল।
তো আমি তো ওই জন্য জুমের লিংক না না তারপরে তো আমাদের ছেলেমেয়েরা বেরিয়ে জুমে ঢুকেছে
আচ্ছা আচ্ছা।
আমাদের ছেলেমেয়েরা জুমে আছে এইখানে বাইরেররা আছে আবার ইউটিউবেও চলছে 3 জায়গায় চলছে তো ওই জন্য এইখানে তারপরে কম
দাঁড়াও, আমি
হয়ে গেছে
সুপ্রতিমদাকে একটু ইয়ে করে দিই, আমি লিভ করলাম। হ্যাঁ
হ্যাঁ হ্যাঁ সবাই লিভ করো